This video is about the cell cycle. You can think about the cell cycle as the lifespan of a cell. So from the time it's a little baby cell to the time it's an all grown up cell and ready to reproduce. So if we think about the cell cycle as a wheel or a circle, we're going to start right here at this point on the circle, and we're going to work our way around like this, back to the start again. Okay? The first thing that happens to a cell when it's a little baby cell right here, if we follow the arrows, is it starts to grow. Okay? It's got to get bigger first. And then if we continue around and follow the arrows, its DNA replicates. This means that the DNA makes a copy of itself. So there's twice as much DNA in the cell at this point. And then we continue to follow the arrows, and it prepares for something called mitosis. So this whole part that we just went over, so from the green to the yellow to the blue, all of that is a step called interphase. So we're just going to say the first step of the cell cycle is interphase. And interphase has those three parts. The cell grows, the DNA copies, so we get another copy of the DNA, so there's twice as much DNA in the cell, and it prepares for mitosis. And what we're going to talk about with this preparation stage is something called centrioles. So we're just going to say it makes centrioles, and you'll see in a little bit what those are. Okay, the second phase then, kind of highlight this in red. So we're at the end of the blue section. Now we're into this slice of the pie. This whole thing is called mitosis. So step 2 of the cell cycle is mitosis. And mitosis, as you can see, has 1, 2, 3, 4 distinct steps. Prophase, metaphase, anaphase, telophase. And I'll talk about those in great detail in a minute. But basically what's happening in mitosis is the nucleus of the cell divides. Okay, so remember we doubled that DNA back in that yellow part of interphase. Now we're going to split that DNA in half again. And then the last step of the cell cycle is this little section right here. Okay, this is called cytokinesis. Cytokinesis is its own step. It is not part of mitosis. And what happens in cytokinesis is the cytoplasm, remember that's the filling of the cell, divides. Okay, so we've got 3 distinct steps. Interphase, cell grows, makes copies of the DNA, and makes these centrioles. Mitosis, 4 steps, prophase, metaphase, anaphase, telophase, causes the nucleus to divide. And then the final one, number 3, the cytokinesis, divides the cytoplasm. And then we're back to the beginning, back to this baby cell that I talked about earlier. Except now instead of having 1 baby cell, we've got 2 baby cells because the cell just split in half. And those 2 baby cells will both go through growth, copy their DNA, make some centrioles, and then get ready to divide again through mitosis. Okay, so we're going to look a little more closely now at that second phase called mitosis in this next set of slides. And in class, you're actually going to be using some string and some beads and some other little tokens to show mitosis and describe mitosis. And that's what your assessment for this particular learning target is going to be, is can you show and describe these steps of mitosis? So what you're going to need when you get into class is you're going to need a big string, a little string, 3 sets of beads. So this is 1 set of beads and then its partner. And here's another bead and its partner, and the third one and its partner. Okay, and this will make more sense when you get into class. But 3 different colors of beads in sets. And then finally you're going to need these 2 little tiles. Okay, and you'll be doing different things with them to show how mitosis works. Mitosis has those 4 phases that we talked about earlier, prophase, metaphase, anaphase, telophase. So you'll be showing each of those phases with the supplies I just talked about, and you'll also be describing them. And then finally, we'll wind up with cytokinesis as well, kind of include that with the telophase stage. Okay, let's start with prophase. What you're going to have to do with prophase is set up the cell and describe it. And you're going to have to use some particular words when you describe what's happening to the cell. So the first thing you're going to do is you're going to have your cell all set up with the big string and the little string. And you're going to say, let's see, there's your different colors. I'll just show them like little sticks. Those are your colored beads in the middle. Okay, those are your chromosomes. So you're also going to have these tokens out there. Those are the centrioles. Okay, so I just used two of your words. We got chromosomes, again here in the middle, and the centrioles are these guys on the side. The nuclear membrane is this one in the middle, and the cell membrane, of course, is around the edge. So in prophase, three things are going to happen, and you're going to describe those for me as you show them. So the first thing that happens is the nuclear membrane disappears. So you're going to take that little string out of the cell. So all you've got left in the middle are some chromosomes. The second thing that happens is that the chromosomes become more visible. So you're going to separate the chromosomes so they're not just like a pile of chromosomes sitting in the middle, but that they're more distinct. So they'll kind of be like this. Remember, you have 6 total. Okay? And then finally, the centrioles move to opposite sides of the cell. So if this centriole right here is there, the other one's going to move opposite of it, so it ends up over there. Color that in. Get rid of the other one. There. So now the centrioles are on opposite side of the cells. So again, 3 things happen in prophase. The nuclear membrane disappears. The chromosomes become visible, and the centrioles move to opposite sides of the cell. The next stage is metaphase. So we need to describe the cell again, set it up what it should look like, and do our keywords. So outer cell membrane, nucleus is gone. Remember, we got rid of that. Our chromosomes are kind of scattered all over. 2, 3, 4, 5, 6 of those. We had our centrioles over on the side. Just do those all in red now. Okay? And now we're going to say what happens in metaphase. So in metaphase, these little centrioles that we drew, these little red blocks, shoot out these things that kind of look like spider webs. And you're just going to describe those for me. You're going to say that the centrioles shoot out the spindle fibers. Those are the spider web type things. And what those spider web type things do is they hook onto the chromosomes and they move them to the center of the cell. So I'm going to erase these just so you can see what they would look like after they're done moving. When you do this in class, you'll actually just physically move them. So I'm going to have all 6 chromosomes now lined up down the middle of the cell. Okay, so I've used the 2 words, spindle fibers, and the middle of the cell is the cell equator. So in metaphase, again, all that happens is the spindle fibers shoot out from the centrioles, attach to the chromosomes, and move them to the middle of the cell. I like to remember metaphase, M equals middle. That helps to remember, whoops, what happens there. So the M in metaphase is for the M in middle. Next step in mitosis is anaphase. So again, let's describe the cell here. No nucleus yet, we just have the cell membrane. We just had all of our chromosomes lined up down the middle and our 2 centrioles over here. Right, that's where we were. And we had the spindle fibers holding those chromosomes kind of in the middle. Okay, so in anaphase, what's going to happen is the spindle fibers are going to begin to shorten. They're going to begin to pull back. So I'm just going to erase a little to show that they're shortening. And as they shorten, they split the chromosomes apart so that half of each chromosome goes in each direction. Okay, so I'm going to erase these chromosomes quick to show what this would look like. So we had one chromosome here at the top, half of it goes this way, half of it goes that way. Okay, they split up and they get pulled in opposite directions. The place where they are broken apart is called a centromere. So if our chromosome actually looks like this, and you'll see this better in class, right here in the middle is where that centromere is. So the spindle fiber will attach to the centromere from either side, and when it pulls back, I'll show that here too, it's, like I said, pulling them apart. So it breaks the centromere. So in anaphase, again, the main thing you want to say is that the spindle fibers pull back, breaking apart the chromosomes at the centromere, and it pulls them to opposite sides of the cell, toward those centrioles. Telophase and cytokinesis. Remember, this is part of mitosis, this telophase. And then we're just going to tack cytokinesis onto the end because it kind of all happens at the same time. So we did have a cell, again, that looked like this, with our chromosomes kind of over on the side, and our centrioles here, okay, and our little spindle fibers, we're holding them there. Okay, in telophase, what's going to happen is these chromosomes are going to kind of bunch up on either side of the cell, and the centrioles will disappear along with the spindle fibers. So everything here goes away, and to show that the chromosomes are bunching up, I'm just going to kind of erase and then add back to it. So our chromosomes that were over here are now in a pile, and our chromosomes that were over here are now in a pile. Okay, so we have two distinct areas where the chromosomes are. And what happens then is that nuclear membrane that we had before reforms around each set of chromosomes. Okay, remember we started with a cell like this with the nucleus in the middle and the chromosomes inside. Now we've got a cell with two nuclei, so that means there's two nuclear membranes and two sets of chromosomes. Remember I said the centrioles disappeared, the nuclear membrane comes back, and now finally the cell membrane is going to start to pinch in. So that would look like this. You're going to take that string, you're going to start to drag it in like that, and continue dragging it in until it completely divides the one cell into two. So completely pinches apart. So now you have two new cells. Each one looks like the original cell with the same number of chromosomes in it, but just a little bit smaller. But remember, the next step of the cell cycle is actually to grow the cell bigger again so it can do mitosis all over and over. So the cell is making a copy of itself by first copying the DNA and then splitting that DNA in half, and then finally splitting the cytoplasm and the membrane in half with cytokinesis. Again, you'll be doing this in class with some materials, and so you'll have to talk through each of these stages using these words, and it'll get real familiar to you after a little bit of practice.